Okay, welcome to today's ASBMB transcription webinars. The monthly transcription webinars aim to promote collaboration and knowledge sharing among scientists interested in the general areas of transcription, chromatin, and epigenetics. Today we have two outstanding speakers. Each speaker will talk for about 20 to 25 minutes, followed by answering questions for 5 to 10 minutes. After both talks, I will open two breakout rooms, one for each speaker to answer additional questions. Today, we are fortunate to have Dr. Greg Wang from Duke University to co-host with me. So the audience, when you ask questions, please raise your hand. You may also ask questions in the chat box. Our first speaker is Professor Anders Hansen from MIT. The title of his talk is Mechanisms of Loop Range Enhancer Promoter Looping. So welcome, Anders.
Okay. First of all, thank you so much for having me, and I'm excited to share some of our recent work here with you today. Can you all see my screen? Okay.
Yeah.
Okay, great. Yeah, so I'll talk about some unpublished and unpreprinted work we're doing, trying to understand the mechanisms of enhancer promoter looping, and I would love your feedback on it. Since this is a focused transcription meeting, I'm going to give a relatively minimal introduction, um, and uh, and just say gene expression regulation is important. Enhancers are key units of gene control in mammals. And because enhancers can be located very far away from the gene they control, the key question that we're trying to address is how does the cell make sure that the right enhancer loops to and activates the right gene? What are the rules that determine whether this enhancer is going to loop into the first gene or loop into the second gene? And over the last many years, we've done lots of studies focused on this question. But today, I want to focus specifically on what looping actually means. We, as a field, use the word looping, but what does it actually mechanistically mean in space and time? So enhancers were discovered in 1981. They are the primary units of gene control in mammals, but there's still questions about how they actually interact with their target gene. The classic model is the stable contact model, where there's a stable contact loop between the enhancer and the promoter that allows activators at the enhancer to directly activate the promoter. But work from us and others, including Sabate et al., Luca Giorgetti's lab, Thomas Greger, several other people have suggested that chromatin loops are not stable. And so you could imagine modifying this model to a dynamic contact model where the mechanism is still contact between the enhancer and the promoter, but the contact is transient instead of stable. At the same time, work from lots of people over the last, especially 6-7 years, have suggested that when enhancers activate genes, they're actually separated by 200-300 nanometers. And this had led to several action at a distance models. One idea is a condensate model where a condensate bridges between the enhancer and the promoter across, say, a few hundred nanometers, such that you don't actually need the enhancer to contact the promoter. The condensate holds it together. And this is just one example of action at distance. There are also other models involving gradients of transcription factors, gradients of RNAs. There's poly-ADP ribosylation models. There's really a lot of different mechanisms you could imagine to explain action at a distance. And you can imagine action at a distance being stable or dynamic as well. And so what I'd like to do today is try to distinguish between these four models. And if you want to distinguish between things that are stable and things that are transient, we need high temporal resolution. And if you want to distinguish between things that happen on the order of 30 nanometers from things that happen on the order of 300 nanometers, we need very high spatial resolution. And if we want to do everything, we need everything. So we need high temporal and high spatial resolution. And so today I'll share with you a system we've built to do this, where we can simultaneously look at enhancer promoter distances and nascent transcription in real time and see, is there a dip in enhancer promoter proximity before you get a burst of transcription? So today I'll be focused on basically a very straightforward question, which is how close does an enhancer need to get in space and for how long in order to activate a gene? And I'll start by telling you about a synthetic biology platform we've built specifically for this purpose. And then I'll give you 5 different approaches to estimating how close and for how long. And I won't go through the details of the 5 estimates right now. I'll go through them in a little bit. but just to say that they surprisingly perhaps end up converging at exactly the same numbers. But first, I need to introduce the team. So this has been a 3-lab collaboration, a very close collaboration with Christoph Seckner's lab and Leonid Mirny's lab. We've been collaborating for many years. And there's also been key contributions from Miles, Christos, James, and Jakob. And it's been led by Harvey, Henrik, and Jack. Okay, and it's been a very interdisciplinary collaboration. So first, I'll talk about the synthetic biology platform we've built to study this. And so just to motivate this, dynamic or transient contact is actually technically very, very difficult to detect, because if the contact is very brief and there's temporal delay between induction of transcription and RNA appearance, you might miss the interaction. At the same time, there was really a paper a few years ago from Alexander et al. I think really changed how the field thought about enhancer-promoter interactions, because they looked at live tracking as really a landmark breakthrough paper in the field, technically extremely sophisticated. They looked at enhancer-promoter interactions in real time in living cells and nascent RNA, and then they look at all the MS2 bursts, and they pile up on the EP distances before the MS2 burst, and they see there's basically no dip, and the distances are 300 nanometers. And so that led them to rule out contact, and they say supports an unexpected mechanism for enhancer control of SOX2 expression that uncouples transcription from enhancer proximity. And so this of course motivated action at distance mechanisms. And so one of the first things we did was we did some 3D polymer simulations to ask, okay, if there is transient contact, what will it take to detect it? And we made a list of 5 key things that are necessary. So I want to just introduce the system we built. So we built an enhancer promoter pair in an empty slate on the genome. So this is a part of the genome where there, for lack of a better word, there's not a whole lot going on. It's as close to a blank slate as we can get. There's one CDCF site that we've deleted. And then we inserted a synthetic enhancer and promoter. And we built a synthetic enhancer by going to Rick Young's super enhancer paper. And we found 3 small super enhancer fragments here that we Frankenstein together to make a a synthetic enhancer that's 1.9 kilobases. For our promoter, we used the HSP68 regulatory sensor that is a minimal promoter that doesn't have activity on its own, but it's responsive to distal enhancers, and it's been used very effectively by Francois Spitz and several others over the years. And then this promoter drives both BFP and MS2. Okay, and we place them 339 kilobases apart in the genome. Then we move the enhancer around. So if the default expression is 339 kb, when we remove the enhancer, expression goes to 0, which is good. The promoter is entirely responsive to the enhancer. And then we can move the enhancer closer and closer and closer, and we get a monotonically increasing expression level as we would expect. And when the enhancer is right next to the promoter, we have 30x higher expression. So that seems to suggest the system is working. We also did region capture micro-C, and as expected, when we just have the fluorescent labels, we basically don't have any structure in this blank slate. But when we insert both the enhancer and the promoter, we see the formation of a dot in the region capture micro-C map indicating a loop. So we see a structural enhancer promoter loop, as we would expect if there was a functional interaction between the two. Next, we want to track things. And so here's a polymer simulation of 2 segments on a polymer, and then we're tracking the 3D distances as a function of time. This is simulated data. So if you want to do this very precisely, we need bright, small labels. Classically, we and others have used fluorescent repressor operator systems like LAGO, TED-O, and QMate, and they work well, but they unfortunately tend to be very large if you want high localization precision. And so here we use instead the Paris systems. The Anchor 3 system from Kerstin Bustecki has been developed and works very well. And to have another pair, an orthogonal pair, we developed a novel one that we call SYN BSR1 that also works very well. And these are very attractive because we can get 500 fluorescent proteins over 1 kb without perturbing endogenous regulation. So now we can label the DNA labels. What about RNA? So here we use the classic MS2 MCP system that was developed by Bertrand and Rob Singer back in the day. What happens is that you put these MS2 hairpins into, in this case, the first intron of the gene, And then when polymerase transcribes it, you have these hairpins, you have fluorophores bind the hairpins, and you see a bright dot. And so here's a simulation of the MS2 intensity signal would look like. So you have a bright signal on the gene until the RNA dissociates, and then it goes away. And then this is what it looks like in real life. So we're using diploid mouse and embryonic stem cells. So you can see there are two alleles. The yellow allele at the bottom is already bursting. And then when I play the movie, you'll see the top allele in white with a little bit of a delay starts bursting as well, right around now. So we can measure nascent transcription in real time in living cells. And now we turn to super resolution live cell imaging, and we want large fields of view, and we want isotropic and high resolution. So we use lattice light sheet mediated super resolution live cell imaging, and this allows us to get these very large fields of view. So mouse embryonic stem cells grow in these 3D colonies. So you can see here we're imaging two 3D colonies. We have two pairs of dots per cell. So here you see the enhancer promoter pair here, and here you see the enhancer only, and here you see transcription. Maybe, I don't know how well it shows up on Zoom, but there's little flickers here. So there's a burst, there's a burst. Here's another burst. Here's another burst. So we can track the bursts in real time. Of course, these movies are gigantic, so we developed an image processing framework. This is a Herculean task because we have on the order of 100 terabytes of data. For reasons of time, I won't go into it, but just to say we have this framework here that allows us to segment the nucleus and then make individual trajectories. And so here's an example movie. So we do 3-color imaging. So we have the enhancer, the promoter, and nascent transcription. We do 3D imaging, but the screen is 2-dimensional. So I'm showing each nucleus in projection. So here's an XY projection, XZ projection, and YZ projection. And then we're tracking this enhancer and this promoter. And you can see at the beginning of the movie, there's no burst. And then when I play the movie, you can see we're tracking things over time. And this, I want to emphasize that the trajectory I've picked here is extremely cherry-picked and unrepresentative. Basically, it's the only trajectory that looks like this in the entire data set. But I chose it because you can see there's flickers at long distances. And then there's the longest loop we've ever observed over the years. It lasts on the order of 2.5 hours. And the reason why I chose it is because you don't need any sophisticated analysis to see here you have a loop at short distances for a long time that then eventually breaks and the distances shoot up. And this coincides with a gigantic burst of transcription here. So I'll just let it replay again. So you see the loop forms around here. So now the loop is formed, and now we're going to see a gigantic burst of transcription that you see down here in blue. And I, again, just want to emphasize this is an outlier, just for illustration purposes. Most of the CDCF loops in this system last on the order of 20 minutes, consistent with prior work. Here's another example where what I showed you was a 6-hour movie. Here I'm showing you a 1-hour movie at higher frame rates. And again, we can track EP distances with high precision, and we can see bursts as they appear throughout the movies. Okay, so we've built this synthetic biology system that we think is optimized for distinguishing enhancer-promoter interaction mechanisms. What I will do next is go through 5 orthogonal ways of estimating how close enhancers and promoters have to get in order to activate. And the first few estimates will be a little bit indirect, and then we'll get more and more direct. But the first thing we started with was a simple 3D structure-function relationship. So here are 7 of the many cell lines we built. So the default cell line has the enhancer and the promoter 339 kb apart. If you put CDCF sites at both the enhancer and the promoter, expression goes up almost by a factor of 20. At the enhancer only or the promoter only goes up by a factor of 3, and we can also change the difference. And then for all of these, we performed region capture micro C. And so here are the different region capture micro C maps. And then we take the strength of each of these loops, each of these dots, and put them on the x-axis, and then we put expression on the y-axis, and we actually get a straight line. We get a basically perfectly linear relationship. Here I'm showing the linear relationship when the readout is BFP, and here I'm showing the relationship when the readout is MS2 nascent RNA. So, and also a nascent RNA and BFP correlate as well. So you get a perfectly linear relationship. And if you think about it, this can actually only occur under quite unique circumstances. So there are two key thresholds here or radiuses. There's the REP radius, which is how close does the enhancer and the promoter have to get in order to activate. But then all chromosome conformation capture assays like Hi-C and Micro-C and 3C, they also have a capture radius. You only get proximity ligation below a certain capture radius. And it turns out that you can only get a linear relationship if the capture radius of the 3C assay matches the functional interaction radius between the enhancer and the promoter. If one is bigger than the other, you get a nonlinear relationship that's either convex or concave. So the fact that we get a perfectly linear relationship suggests that these two are the same. And Joe Paggi and Bin Zhang has previously estimated the interaction radius to be on the order of 42 nanometers for micro-C. We performed our own polymer simulations. Our estimate is a little bit lower at 25 nanometers, but as you can see, our 95% confidence intervals are wide. So it's on the order of 25 to 42 nanometers here. And we can basically only explain this if the interaction radius and the capture radius are similar. So estimate 1 lands on the order of 25 to 42 nanometers. Let's go to estimate number 2, which is also a little bit indirect, which is how much does adding CDCF sites boost expression? So as I showed you, if we have the default cell line and we add CDCF sites to both the enhancer and the promoter, expression goes up by almost a factor of 20. But the 3D distances actually don't change very much. So if you see here, the median distance goes from on the order of 250 nanometers to on the order of 170 nanometers. So the distances only change a little bit. And so if you think about it, hypothetically speaking, suppose the functional enhancer-promoter interaction radius was 250 nanometers. Well, then the cell line without CDCF sites is in contact half the time, 50% of the time. And so the maximum you can boost expression is to go to 100%. And so the maximum you can boost expression by adding CDCF sites is a factor of 2. But we here observe a factor of 20. And so to see how much how low the functional interaction radius needs to be to explain this, we perform 3D polymer simulations, and we end up with an estimate on the order of 23 nanometers. So this estimate really suggests a contact mechanism where we can only explain the dramatic increase in expression from CDCF sites if the functional interaction radius is very, very small. Okay, so our estimate number 2 places the functional interaction radius on the order of 21 to 24 nanometers. Let's go to the first 2 estimates here have been relatively indirect. So let's go to a more direct estimate. So as I mentioned, in the SOX2 system, there was no correlation between the onset of a burst and EP proximity. So we wanted to basically do this analysis and to see if in our system there is such a relationship. And here's another trajectory where you can see we have lots of bursts. And so we can basically go to the beginning of every single burst, and then we can ask, was there a dip in the 3D distances? But it's important to keep in mind that EP, even in a dynamic contact model, you're not going to have instantaneous RNA after EP contact, right? Because you need to recruit activators, chromatin remodelers, maybe post-release factors, etc. And then polymerase has to transcribe about a kilobase or so before the MS2 begins to appear, and then binding proteins have to bind. So there's going to be a little bit of a delay between contact and transcription. And so to capture this, we used a 2-layer long short-term memory model, which is a recurrent neural network that's ideally suited to these sort of memory effects. And we use change point detection. We use change point detection to capture the beginning of all the bursts. And then we ask, what do the EP distances look like before the onsets of the bursts? And we actually see a very clear dip, both in the 30-second data and in the 5-second data. The dip is on the order of 120 or so nanometers here. And it lasts on the order of 10 to 15 seconds. And after the dip, we see a big increase in expression. So 120 nanometers or so sounds like more than contact, but it's important to keep in mind that measured 3D distances will always be much bigger than true 3D distances, right? If you have 2 things that are in contact and you add uncertainty in X, in Y, in Z, suddenly the measured distance is much bigger. And so to account for that, we use 2 different approaches. One is an experimental control. We can put the 2 fluorophores as close together as they would be with contact. And we have 2 versions of this. one where we just put them right next to each other with 400 base pairs apart, and one where we put the enhancer in the middle. And in both cases, the histogram of 3D distances in the proximal label controls actually matched the histogram of 3D distances before burst onset. So burst onset looks like proximal label controls, which we interpret as qualitatively as physical content. The other approach is to use simulations. So we can say, okay, let's do experimentally constrained 3D polymer simulations. We have estimated how much noise we have in our data, chromatic aberration errors, etc. So let's add noise to our simulated data without noise, and then let's ask what would be the functional interaction radius in our simulated data that would match the experimental data. And if we do that, we get to an estimate of 44 nanometers. So either qualitatively contact or 44 nanometers here. So here we arrive at contact to 44 nanometers for our third estimate. And we also have the first estimate of how long do enhancer-promoter interactions actually last. And I want to emphasize here that there's significant uncertainty in this estimate, but it's on the order of seconds to tens of seconds. So it is much more than sort of random diffusive contact. It's stabilized, but it's also much, much less than CDCF loops, which last on the order of 20 minutes on average. Okay, so we now have our third estimate, which lands on the order of contact to 44 nanometers and on the order of 10 seconds. So now I'll move to our fourth estimate, which is directly using Bayesian inference. This estimate, I should say, this part is still a little bit in progress. So we're still sorting out a few things. So the numbers for this part here might change a little bit. It's by far the most direct and rigorous way of analyzing the data, but it's also the most complicated. So I won't be able to explain it in full detail. But the basic idea is that we have polymer dynamics that eventually turn on the promoter that leads to polymerase loadings and MS2 observations that result in MS2 trajectories. And this is what happens biologically. And then what inference does is that it tries to go the other way around and say, start with the output and then try to estimate what the input would be. And we've been working on this for 3 years or so. It is relatively involved mathematically. But the basic idea is that we have noisy observations of the data for EP proximity and for MS2. And then we can use the inference to reconstruct the most likely samples. And we can add confidence intervals. And then we can take this data and we can ask, okay, for different interaction radiuses, what would this look like? So here we have 50, 100, and 200 nanometers. So you can see if you have 200 nanometers, you're actually in quote unquote contact most of the time, or at least you are above, the probability of contact is more than half most of the time. Whereas if the interaction radius is very small, you're very rarely in contact. And then we can ask, which one of these best explain the observed bursts? And again, I put a caution work in progress sign here because there's still a couple of kinks we're sorting out here. But the estimate for this comes to be on the order of 45 nanometers or so. So we are so again, it's similar to the other estimates. Okay, so for our fourth estimate, we land on the order of 45 nanometers. Now I'll move to our fifth and final estimate, and then I'll wrap up. And so here we so far all the estimates are consistent with a dynamic contact model where the enhancer and the promoter need to get within 20 to 40, 50 nanometers in order to activate, and where the interactions last seconds to tens of seconds. And you'll notice that the interaction radius is actually strikingly similar to the size of the cohesin complex, which is also on the order of 40 to 50 nanometers. So one prediction of dynamic contact is that since cohesin has the right size and is expected to make contacts that last a few seconds, cohesin-mediated contacts should be special and exert a very strong effect on transcription. And one way to test this is to remove cohesin. So since it's essential, we use acute degrons where we can deplete cohesin for 2 hours. And then we actually see almost complete loss of bursting within 2 hours. So here's just an example for the 87 kb gene, where you see lots of bursts here initially. And then after cohesin depletion, they more or less all go away. Here's the one with 339. Again, you see lots of bursts that mostly go away. We see on the order of an 80 to 96% reduction in transcription, but actually for the cell lines where we have 339 kb between the enhancer and the promoter, it goes down to the point where it's statistically indistinguishable from basically deleting the enhancer and also deleting the promoter. So cohesin really seems to be special, which again is consistent with a dynamic contact model. Next, we turn to insulation. So if cohesin is special, one way of removing cohesin-mediated contacts is to add CDCF insulators. And CDCF insulators should basically only work if the functional interaction radius is small. Because if the functional interaction radius is small, then it blocks extrusive interactions. But if the functional interaction radius is large, it doesn't really matter because the functional interaction radius is so big that it doesn't really care about whether you put some CDCF sites in the middle. And so if the functional interaction radius is small, we should see a strong reduction in transcription by adding these CDCF sites. And that's indeed what we see. We see actually a 97% reduction in transcription. So it's a very, very strong loss of transcription when we add CDCF sites here in the middle, pointing in both directions. Next, we repeated this experiment where we have the CDCF sites already at the enhancer and the promoter, and then we put convergent CDCF sites in the middle. And here, the predictions are a little bit less clear. So one model that has been proposed is a loop stacking model or boundary stacking model. According to this model, adding CDCF sites in the middle should actually increase expression because now you just need to make two small loops and you form two stacked boundaries. And this is also called a rosette. And so the loop stacking boundary stacking model would predict that expression should go up. And if you have a large interaction radius, it shouldn't really matter very much because you're going to be within contact no matter what. Only if the interaction radius is very small would you expect this to reduce expression. And that's actually what we see. We see a 90% reduction in expression here. So this argues against this sort of loop stacking, boundary stacking picture being positive for transcription and is consistent with a very small interaction radius. How small is, of course, an important question. And we perform polymer simulations to estimate this constrained by experiments. And we actually find that no matter how small we make the interaction radius, we can't explain this gigantic drop in transcription without adding one additional aspect to the model, which is time gating. And so time gating has been, time gating, the idea is that the enhancer and the promoter will filter out contacts that are too transient. And so if they're very, very transient, they don't lead to expression, they need to last on the order of 2 seconds or so in order to activate. And many people have looked at time gating before in both yeast and human cells. There's also a nice recent preprint from Luca Giorgetti's lab looking at this. And so there it's all consistent. And we estimate the time gate to be on the order of 2 seconds. And our best estimate of REP from this is on the order of 25 to 55 nanometers. So to summarize and finish up, we've built a synthetic biology platform that allows us to study enhancer-promoter interactions without redundancy in a clean locus with very high spatiotemporal resolution. By looking at the structure-function relationship between region capture, micro-C, and expression, we estimate the interaction radius to be on the order of 25 to 42 nanometers. By looking at how much CDCF boost expression, we end up on the order of 21 to 24 nanometers. By applying machine learning to the imaging data, we end up at contact to 44 nanometers, and we estimate that the interaction timescale is on the order of 10 seconds, though there's significant uncertainty here. Using a Bayesian inference approach, we end up on the order of 45 nanometers, and by doing perturbations to cohesin and CDCF sites, we end up on the order of 25 to 55. And so coming back to the motivation, there are various action at a distance models that have been proposed, but the data, at least for this EP pair, is consistent with dynamic contact. And so every single one of our estimates have different uncertainties, but they all line up to be on the order of 20 to 50 nanometers. And our consensus estimate is on the order of 35, which is much less than some observations of 200 to 300 nanometers. So we think the data is consistent with a dynamic contact model for enhancer promoter interactions, where we have stabilized interactions. They are much more transient than CDCF loops that last 20 minutes. They last on the order of seconds to tens of seconds. And we also need to incorporate time gating, where two transient EP interactions get filtered out, and only if they last at least a few seconds do they lead to transcription. And this might be particularly important for EP selectivity. We are fortunate at MIT to have one of the world leading structural biology of transcription and genome organization experts, which is Seychelle Voss. So we asked Seychelle, what is a likely macromolecular complex that mediates enhancer promoter interactions? And she thinks it could be activator precipitated mediator dimerization, in which case it would look something like this. And the shortest end-to-end distance would be on the order of 39 nanometers, which is very similar to our consensus estimate of 35 nanometers. I want to be very clear, our data does not allow us to say in detail what are the macromolecular transcriptional complexes that bridge enhancer promoter interactions, but the distances are consistent with what you might expect from structural biology. So to end, I just want to end also with some limitations here. So of course, generality remains to be determined. We've looked at one EP pair in mouse embryonic stem cells. It's completely possible and perhaps extremely likely that other EP pairs in other systems will behave differently from the one we studied. The other thing I should say is that the original model for condensate formation at super enhancers is for these very long super stretch enhancers. And so there, maybe you have 25 kb and you have many sub enhancers, and then they cooperate to make a condensate. And that our data does not rule that out at all. Our data is completely consistent with super stretch enhancers forming condensates. For practical reasons, we wanted to study a very compact enhancer, so our data cannot say anything about these long super stretch enhancers. The other thing I'll say is that dynamic contact is extremely transient, is extremely difficult to see because it is so transient. And so we think that dynamic contact is actually consistent with previous studies as well. We also think that if action at a distance, if that was the general mechanism for most enhancers, selectivity would be difficult to explain. The latest estimate from ENCODE is that we have on the order of 2 million enhancers in the genome. So the nucleus is basically full of enhancers. And if you can activate across 300 nanometers, on average, you would be in contact with on the order of 40 genes or so. The timescales are more uncertain. We estimate here seconds to a few tenths of seconds. Our best estimate is on the order of 10 seconds or so, but there's uncertainty there. And we hope this technology will help study future EP pairs as well. So I want to thank my lab and especially thank the team here. So again, I want to strongly emphasize this was a 3-lab collaboration with Christoph Seckner's lab and Leonid Meni's lab. Very close collaboration. Really want to shout out Harvey, Henrik, and Jack, who led all of this. And also just to say, we're still working out a few things, and especially the Bayesian inference, there's still a little bit of uncertainty. So I put a caution work in progress sign here to reflect that, but I'd be happy to take any questions if time allows.
Yeah, great talk, Anders. We have time, I guess, for one question from the audience. Anyone would like to ask a question? So, okay, I see one raising hand. So yeah, go ahead.
Oh, hi. Can you hear me?
Yeah.
Very interesting talk. So I know that you pointed out that your system is only targeting to one pair, one enhancer, one promoter study. So, however, in your loop extrusion and the isolation system, if you realistic, if you put your CTCF in a gene that they may have multiple enhancer contribute to one, gene promoter activation. Whether you put, you put in the CTCF in solution, they will make this promoter to pick another enhancer, or whether that enhancer dynamic will be changed in your system, and potentially.
Oh, I see. So if I understand correctly, you're saying when you put in the CDCF insulator, could the promoter now start talking to another enhancer, not the one we
Right,
put
right.
in?
Yeah,
Yeah,
yeah.
no, that's a good question. Yeah, so that is one of the challenges with studying this is that there's so much redundancy in the genome. The genome is full of stuff, but so we tried to circumvent this by picking basically an empty region. So we don't have the RNA-seq track here, but we put the enhancer and the promoter around here, and there aren't really any other active elements nearby, and the ones that were there we deleted. So we think this is as close as we can get to a blank slate, and we also know that when we delete the one enhancer, then expression basically goes to zero. So of course we can never 100% exclude this, but we think it's unlikely.
Okay, great. So I'm sure the audience will have more questions in the breakout room session. Next, my colleague, Professor Greg Wang, will introduce second speaker.
Sorry. Can you hear me?
Yeah.
So now we're gonna have the second outstanding speaker, Shark Coit from Novaya University from Canada, and his seminar title would be acetotransferase complexes in epigenetic mechanism linked to gene regulation and oncogenesis. Shark, please go ahead.
We see your slides. Yeah.
I think Jack is muted.
Oh.
Shark, can you unmute yourself?
Okay, let me.
We can see your slides, and you just need to unmute. Yeah, I see you unmute yourself.
Yeah, right. I was not seeing the button anymore.
Wonderful.
Yeah. So why is it not starting now?
So if you unselected the pointer, then you can click that.
Okay.
Yeah. Well, you have to unselect the pointer and start the slideshow.
How do you unselect it?
Try escape. Yeah. Do
exactly.
that. Yes.
Yeah, that's
great.
I cannot believe, after so many years, we're still struggling. All right. So thanks so much. And again, these webinars are so exciting, interesting, and I'm very honored to be participating in that. So I'll talk today about acetotransferase complexes. I know that last webinar you had Asifa Akhtar that talked about the specific acetotransferase complex. I'm going to talk about a couple of different ones. So as you all know, the epigenome has been characterized very thoroughly in terms of where the different histone marks or histone variants are present in genome compared to heterochromatin, silenced chromatin by polycomb, and active gene and enhancer. I won't argue if there's like 35 nanometer between that enhancer and that promoter, but now we know. So an acetylation, of course, is located more in the active promoter at active enhancer region. But a characteristic that I want to underline here is that the protein that are depositing or changing those chromatin composition, depositing marks are most of the time part of large multi-subunit complex. And those multi-subunit complex can have the activities of depositing a mark or removing a mark, but also most of the time contains like subunit that have reader domain that will recognize other marks in the chromatin and allowing the epigenetic crosstalk in different location in the genome, which is also quite.
I was going to pass today, like just doesn't have cells.
All right. So my lab has been focusing our studies on a family of histone acetyltransferase complex, the MIST family of histone acetyltransferase complex. There's six different complexes. You heard from Asifa about the MOF complex, which is a small tetrameric responsible for all of H4K16 acetylation, of course, important in transcription and DNA repair. It's also in another form with the NSL complex, which is much larger, with a change specificity. But my lab has been focusing on U4 TIP60 complex, a very large complex involved in gene regulation, essential for cell viability, but also on those smaller MIST family complex, the Mos-Morph complex, the HBO1 complex, we call it two different forms, having different specificity for acetylation, H4, H3K14, H3K23, but also involved in gene regulation, stem cell nests, and hematopoietic stem cell, embryonic stem cell, neuro stem cells, and also in this case in DNA replication during cell cycle. Important thing is if you look at like these three, like MOS is responsible for all of H3K23 acetylation in the cell, while MOF is all H4K16, and HBO1, this flavor of HBO1 is for all H3K14 acetylation. Those three sites are the most abundant site found in the genome. Between 10 to 20% of the residue is acetylated in the cell. So obviously, we often think about acetylation. Oh, you just need acetylation to open chromatin, activate transcription. But obviously, those lysine specificity mean something. And so acetylation is definitely part of the signature, the epigenetic signature that is specifically read by some reader domain to provoke a biological response and pathway. And if you look, for example, the Mos-Morph complex, there's only 4 subunits, but you look at the domain, the reader domain, and the functional domain found in that complex. So this complex is responsible for all of H3K23 acetylation in the genome. In the knockout of BRPF1, you see you lose completely that acetylation. But it has all those reader domain, a PhD domain for H3K4 methylation at transcription start site, a PWP domain for H3K36 methylation in the coding region of gene, a bromo domain for H4 acetylation, a double PhD domain, finger domain that recognize another acetylation, H3K14 acetylation, and a 2 wing helix domain in the catalytic subunit. And these are quite numerous for such a small complex, but it's also demonstrate the crosstalk there is in vivo in gene regulation and epigenomic regulation. Like if you look at that DPF domain, the double PhD domain that recognizes H3K14. Like I said, H3K14 is dependent on HBO1 acetyltransferase. So if you use a knockout of HBO1, you wipe out H3K14, you don't really touch the bulk of H3K23. But if you look at specific gene, you see that you lose H3K23 at specific gene. So really demonstrating there's a crosstalk between HBO1 acetylation and the Mos-Morph complex at those specific locations. The wing helix domain, we've shown in the previous paper that it seems to bind the diet axis in the unmethylated CpG island in the Leaker region of nucleosome. And you mutate those wing helix, you lose the recruitment of the complex or the binding of the complex at specific genes in vivo, and they're correlated with the loss of H3K23 acetylation. So there's definitely an important crosstalk that happens between those reader domains. And that's why I want to emphasize the need to really use native region or work in the most physiological condition to dissect those pathways, those crosstalk in gene regulation, because some enzyme, they don't acetylate free histone, they only acetylate chromatin. Some enzyme, they are not able to acetylate or modify chromatin. They need to be in a native complex to be able to bind chromatin acetylated. And obviously, the complex that have reader domain needs to recognize specific pre-existing mark on the nucleosome to increase this action. So the substrate specificity will differ if you use different substrate in vitro, like free histone peptides, compared to a native substrate like chromatin. And it also differs if you're using the enzyme outside of his normal context, or outside of his normal protein complex. And this is just an example. I think Asifa mentioned about the MOF being in the MSL and NSL complex having a slightly different specificity. If you look at the HBO1 complex, it's in two flavors. It's very similar complex, but one having a JAD subunit as scaffold, and the other one having a PRPF subunit as scaffold. If you give those two complex free histones, they acetylate H3 and H4 pretty equivalently. But if you give them chromatin substrate, you see that the BRPF HBO1 complex acetylate only H3 in chromatin, and the HBO1 JAD complex acetylate only H4 in chromatin. And that reflects what it does in vivo in the cells. This complex acylating only lies in 5, 8, and 12 in H4, and this complex acylating mostly H3K lies in 14. So keeping that in mind, many years ago, we started using CRISPR to purify the endogenous complex to be sure that we're working with the most physiological assembly, protein assembly. So we target all those four complexes by tagging with CRISPR the endogenous gene, and we purify the complex, and those are shown here in silver stained gel. And well, there was no big surprise. We already knew those complex, and we got pretty much what we knew already from previous experiments. So the TIP16U4, the two-flavor HBO1, and the Morse-Morph complex. We got all the mass spec data, the spectral count, the mass spec purified, 10M affinity purified complex. But we had a surprise. We found also in specific purification, so all the subunit of the MLL1, MLL2 complex, which is an H3K4 methyltransferase, very important for development, but only in a specific association with this specific HBO1 complex with the JAD scaffold subunit that's specific for H4 acetylation. And this is from a soluble extract, so it's not colocalization chromatin. If you do benzonase treatment in the fraction, you don't lose interaction. So it seemed to be a physical interaction in the soluble fraction. Of course, MLL, also trithorax in Drosophila, is very important histone methyltransferase because it's critical for gene regulation opposite to polycomb mediated silencing during development, but also it is implicated in 70% of acute myeloid leukemia in childs and 10% of all adults by translocation, cutting the gene for MLL and fusing it with many subunits of the super elongator complex. So there's been a lot of focused research effort to develop drug to treat those leukemia and using drugs also against the downstream factor like BRD4 and DOT1. And the physical link that we see between HBO1 JAD complex and MLL, of course, led to a few labs developing like an approach to treat those leukemia with inhibitor of HBO1 and so from Mark Dawson originally, but also Aki and other labs. So just to make this part very quite short, so what we found along those work is that the interaction is mediated through the menin subunit, a very well-known subunit of the ML complex, and the JAD subunit of HBO1. And what we saw is that, in fact, this is a physical direct interaction that occurs, and HBO1 bound to chromatin is required to recruit the ML complex to specific location on the OX cluster of genes, and that seemed to be a way to important, and even in natural condition, not only in leukemia with the MLL fusion protein, like shown by Mark Dawson and Aki, but also in physical condition. Now, of course, once you recruit MLL, you have the downstream effect going on, and you also deposit K4 trimethylation. We can recruit other factor down the street. So for the second part, I want to focus on the NEO4 TIP60 complex. Of course, it's a much larger complex, more 17 subunits. Also has many different reader domain in different subunit for methylation, for acetylation, and so forth. It is acetylating more than one histone, acetylating H4, H2A, but also H2A variant, H2Z, H2AX. It also acetylates non-histone protein like P53. And it's also linked to ATP-dependent incorporation of H2EZ in the genome, because there's another enzyme in the complex called EP400 that can load H2EZ in chromatin, replacing normal H2A with H2EZ beside acetylation by TIP60. So of course, acetylation and H2AZ localization is often at the same location in the promoter of active genes. It's TIP60 is important for many as a co-activator for many transcription factors listed here, very well characterized for MYC. Also important DNA damage response, repair, autophagy, apoptosis, but it's also, like I mentioned, essential. So it's essential for cell maintenance and differentiation. When you purify the complex, you get all those subunits, but you also get preloaded H2AZ, H2B dimer in the complex. So it's ready to load H2AZ, H2B dimer and chromatin beside acetylating chromatin through the TIP60 Cat5 subunit. So it's very important in cell physiology, but also in disease. So, for example, Neo4 TIP60 has been shown to be downregulated in Alzheimer model system. Important for neuroplasticity. It's also important for neuronal differentiation. It's mutated in a couple of neurodevelopmental syndrome. Of course, it's critical in cancer, protecting from cancer, but also implicated in cancer because it's co-activator of oncogene as well as tumor suppressor. And we've done a lot of work characterizing fusion protein implicating different subunits of the TIP60 complex with subunit of the polycomb recursive complex 1 or 2 in sarcomas. There's very recurrent translocation leading to sarcoma by those translocation. And also in metabolic syndrome, type 2 diabetes by that recently identified subunit JASF1 that's linked to fat liver disease and so forth. So the complex itself has two activity, the acetylation of H4 and H2A, and also loading of H2AZ. Here, this is an in vitro assay where you have chromatin on beads, dinucleosome in this case, and when you put the complex with the H2AZ, H2B dimer, if you put by itself without ATP, you don't have much happening. And when you add ATP, you start incorporating H2AZ in those nucleosome, and you add acetyl-CoA, leading to acetylation at the same time. You increase that incorporation in those nucleosome in vitro. So problem with those protein, those subunits in TIP60 Neo4 is that a majority of them are essential for viability. So we design a deground system with a DTAG system where we can get rid of the endogenous protein, in this case, the P400 subunit responsible for H2AZ loading in chromatin. And you can, when you do that and you get rid of that subunit, you see that you don't lose H2AZ from chromatin, but you lose H2AZ acetyl from chromatin specifically. You can also complement those deground cell line with a Y type and an ATPase mutant. So the TPase activity required for H2AZ loading in chromatin, the complex stays the same. It even still has the H2AZ, H2B dimer in the complex. Surprisingly, we could see that the H2AZ in the complex, preloading to chromatin, is already acetylated. So it's ready to load an acetylated H2AZ into chromatin. So when you look at the deground ATPase mutant, you see that you have a loss or a decrease of H2AZ incorporation genome-wide in about 1/4 of the sites where H2AZ is detected, but you have much larger effect on H2AZ acetyl detected. So the H2AZ that are lost are in fact acetylated H2AZ region in the genome. And if you look at the effect on gene regulation, so the genes that are downregulated, detected by RNA-seq, are in fact the gene that lose H2AZ and H2AZ acetyl, while the gene that we detect that are upregulated in those conditions don't have any change in H2AZ incorporation. So 2 years ago, we with collaboration, a very fruitful collaboration with Zinling Yang and Eva Nogales in Berkeley, we determined the structure, the cryo-EM structure of the endogenous, so purified endogenously tagged TIP60 Neo4 complex, and we could get a 3.4 angstrom structure The structure was very good, but missing, as is often the case in cryo-EM, two functional modules, the acetyltransferase module itself and the Tintin module also. But we could see the motor, the P400 motor that was responsible for loading H2EZ. We could also see the trap module for transcription transformation, that adapter protein. That's the interface where the transcription factor will interact with the complex to recruit it to a regulatory sequence or a promoter sequence. So what we find in the structure is, in fact, the P400 remodeler, ATPase subunit, is the spine of the entire complex. That's the spine on which all the functional modules are assembled to form the full complex. The trap module interacts in the C-terminus. The catalytic region of the module have all the H2EZ, H2B dimer, and the other accessory subunit. The Tintin module binds an N-terminus, and the acetyltransferase module is anchored to two regions on each side of the motor module. So having the structure, we could use structuring for mutation to get rid of certain part of the complex, using again the Degron system supplemented with Y-type and mutant complex, and we could get rid of the trap recruitment module. And what happened there was a bit surprising, because we saw as many sites that were losing H2EZ and H2EZ acetyl as sites that were gaining H2EZ and H2EZ acetyl. In fact, the one that they were gaining were having less to start with, and the one that were losing were having more H2EZ and H2EZ to start with. And that correlated with transcription, because the genes that are highly expressed were losing expression, and the genes that are lowly expressed were gaining expression, and that was correlated exactly with the appearance or the disappearance of H2EZ and H2EZ acetyl. So at the end, we understood that the fact that you lose the recruitment module in the complex seemed to allow the complex to start acting at other places in the genome, a bit like a titration effect, like we see with polycomb complex. So some location that were active, like the cyclin D1 gene, losing transcription, losing H2EZ, but some other gene that were not active to start with become transcribed and gain H2EZ and H3EZ still, and that's correlate with mislocalization of the complex because of the loss of recruitment mechanism, specific recruitment mechanism. So the cryo-EM structure allowed us to determine that the P400 subunit is the major scaffold in the complex, holding all the functional module, that the complex has high flexibility, because we couldn't detect two module in the complex, and even trap was quite flexible as well. There's also key architectural difference between Neo4 complex and other related remodeler complex that might explain or link to its dual activity, not only being a remodeler or not only being an histone modifier, but being both at the same time. And we demonstrate the function of the trap subunit for recruitment, and also that allows us to correlate mutation associated to disease with the structure of the complex, an important part of the complex. Another mutation that we did that was quite interesting is mutating this part of the complex using, again, the Degron system, because it allowed us to get rid of the AT module. As you can see here from the purification of the complex, you lose the AT module of the complex specifically by mutating some part of P400 here, point mutation. So the complex is there, all the other subunits are there, the remodeler subunit is there, P400 with ATPase activity, the H2EZ, H2B dimer is there, and H2EZ is even still acetylated in that complex, which is surprising, even though that module is not there. But when you look at the genome-wide, you see that you lose again H2EZ acetyl, where you lose H2EZ, and this is quite clear. And again, you lose H2EZ where genes are lose expression, as shown here. H2EZ is lost where genes are repressed, and there's no change where genes are upregulated in the condition of the experiment. So at the end, it seems that Neo4 TIP60 is having H2EZ still, but somehow is able to acetylate it in trans before its association with the complex, because even the complex without the AT module have the acetylated H2EZ associated with it. And this acetylated H2EZ, H2B dimer in the complex is loaded on chromatin as pre-acetylated and likely play a role in the remodeling process of and the function of MUF4 TIP60 in gene activation. The thing that's kind of surprising is that all those other remodeling complexes that have been characterized in the past years have shown that they were the structure bound to nucleosome, but we were never able to get the structure bound to nucleosome for some reason. That was quite surprising. Normally, also linker DNA does not influence the binding of MUF4 TIP60 to nucleosome in vitro. We couldn't get any condition where we could see the nucleosome bound structure. And I just want to shoot out to Carl Wu and Robert Lauder, who had a preprint with result very similar to ours. And their model, which is represented here, is pretty striking, and I like it a lot, and that's why I'm using it. So their model is that, okay, compared to the other complex, would need linker DNA to remodel chromatin and open chromatin. This one doesn't need that because it works through recruitment compared to the other one. So a transcription factor recruit the complex through the trap subunit. And then the neighboring nucleosome is accessible, and all those reader modules in the different subunit that we didn't see in the structure specific for H3K4, H3K36, and so forth, can interact with the nucleosome with specific histone marks. And after that interaction, the ATPase can work and load H2AZ, replacing H2A in the neighboring nucleosome. So I think that's a very possible and solid suggestion. And we started testing that by using the histone exchange assay in vitro when we use different substrate with different modification. And we see that when we use a normal H2A or H2AX nucleosome, we have a little bit of incorporation and present of ATP. But if you have phosphorylated H2X, you have more incorporation. If you have H4 that's pre-acetylated on 4-lysine, it's much better substrate. If H4 acetylated also on N-terminal, it's much better substrate. If H2A is in fact ubiquitinated on N-terminal, then it's not a good substrate. So we continue characterizing those assays and figure out how this can work. But we have this structure, but we're missing two modules that we don't see. Of course, that module is a very important one, and the Tintin module. And those two modules have reader domains. So the Tintin module has a bromo domain and a chromo domain specific for methylation or acetylation. The AT module has a PhD domain for H3K4. So maybe why we didn't see the nucleosome bound structure, it's because those very flexible modules are in fact the one binding nucleosome, and you can resolve that with the cryo-EM. So we tested nucleosome by just gel shift, and we could see, in fact, that the AT module itself can bind nucleosome gel shift. You have a band, retarded band, and if you put acidic OA, it's released. So that's known for acetyltransferase. We tested K4 trimethyl nucleosome. We see a better binding, but not so striking. But the thing with many of the chromatin modifier modeler work through interaction with the nucleosome core acidic patch formed by H2A and H2B nucleosome. So we tested that by using nucleosome with the acidic patch mutant, and we see still binding, a bit less maybe, but strikingly, we could see that the nucleosome that has a mutated acidic patch cannot be acetylated by the AT module. So that's very striking. So really arguing that it's required for the TIP60 acetyltransferase activity. So we purify those native complex having mutation in arginine residue, because binding to acidic patch in nucleosome normally occurs through an arginine anchor, and EPC1 has a known conserved arginine anchor based on yeast work by Song Tan. And so we mutated arginine 7, and we can see that purified complex with that arginine 7 mutated lose acetylation of H4 in vitro in acetyltransferase assay. Surprisingly, H2A or H2EZ acetylation in chromatin doesn't seem to be affected, so we still don't understand that, but certainly indicate that the recognition of one tail versus another doesn't work in the same mechanism pathway. The other module that we looked at is the Tintin module. So this is only 3 subunit module, and this is a nucleosome affinity measurement by time-resolved FRET assays with NCP. And it's done in Song Tan's lab in collaboration with Song Tan. And we could see that if you use nucleosomes that are methylated on K36, which is the target of that chromodomain, you see some binding with an affinity of on the order, I don't see the number here, but a few hundred. And the nucleosome is acidated on lysine 12H4, you see an affinity KD of around 200 and something. But if you have bone modification, then you have a quite high affinity between around 50 nanometer dissociation constant, which is pretty impressive. So we show, in fact, that those 2 modules that we don't see in the structure are most likely responsible for binding nucleosome by the complex, as suggested by the model. And of course, we're continuing work to define the determinant for those 2 modules to bind to nucleosome through cryo-EM analysis and their importance for gene regulation and genome stability in your 4-TIP60 function. So the complex itself is seen very flexible, not only through the trap to recruit by transfer factor, but also by those 2 models, the catalytic model and acid module and the Tintin module to work that way. So just end here. I'm not so got a bit long. Anyway, the work I presented. So the initial work with Moz and HBO1 was done by Sukmin Jiang. The work on the complex the structure and the H2Z loading mostly done by Amel Mameri and Catherine Lachance, and the later work with the AT and Tintin module by Mathieu with the help of Melclef for swapping assays. And of course, the crucial collaborator Eva Nogales, Zhenming Yang for cryo-EM, Song Tan and Sue for the FRET assay and nucleosome, and Tanya and Aki for the original work on HBO1 and Mosmorph. Thank you very much.
Thank you, Shark. Very wonderful talk, as always. The floor is now open for questions. If you have questions, please raise your hand. Jerry.
Jacques.
Hey, Jerry.
So this is very curious that you mutate the hat domain, you still get acetylated H2AZ inserted, right? And so how do you think that works? Do you know what other hat might be involved, or is it acetylated before it gets incorporated in the complex, or what?
Yeah, well, you have the finger on it. Obviously, when you knock down or get rid of TIP60, you lose completely the acetylation of H2EZ. So obviously, it's still dependent on TIP60 on your 4. So the fact that it's acetylated in the complex that has no AT module suggests that it happens before association in trans. We have no idea how that works. Maybe, the main chaperone of H2EZ, like Trematic show now, RNP32E, maybe is presenting it to the complex initially before transfer to the complex. But, that's certainly something we need to figure out and investigate. And that was quite surprising. But
interesting.
Yeah.
Have more questions? So Shark, I have a question about the menin inhibitor, which is very exciting development. So you mentioned the HBO complex and ML complex. They have this association mediated by menin.
Yeah.
I know people reported the menin inhibitor will kick off the ML from the site. Do you know, or have you looked at if the SEM inhibitor will also suppress binding of HB1 complex?
Yeah, obviously that's the experiment that is going on, and that we had a bit trouble making it work because would expect the same thing happening, right? To disrupt by disrupting the menin MLL interaction, you would disrupt the HBO1 MLL interaction, and so forth. We don't have, I mean, we have cooperativity of HBO1 inhibitor with menin inhibitor in killing those leukemic cell, but the biochemistry of it didn't show much effect. So maybe there's a second interaction. I think Aki as a model, it's not the only interaction between MLL and HBO1 that occurs. There would be another one. But at the same time, I mean, we didn't do the right control showing that DAR condition, we really disrupted the MLL menin interaction. And so it's still going on, but that's a very, very important point. And yeah, we're pretty slow at doing that experiment.
Great. A lot more work to do.
Looks like we have run out of time. I'd like to thank both speakers for your truly wonderful talks, and